Plant germplasm is any living collection of plant material that represents the genetic line of an individual or species. Globally, collections are increasing in number and diversity. It's important to understand then why ex situ collections are made and how they will be used in the future.
So there are multiple uses for ex situ germplasm. First and foremost is obviously conservation. We can also use it for ecosystem restoration, plant breeding programs, or for horticultural programs. In places like Kings Park, where are botanic gardens, the germplasm can also be used to grow plants for display, which is an important component of public outreach and education. Depending upon why we collect germplasm in the first place will dictate how we collect it in the field and then what kind of material we collect as well. So for example, when we're collecting for conservation programs, we often need to collect maternal lines from individual plants. But then for other end uses, such as plant breeding, we might be interested in particular forms. So plants with different flowering colour or flowering time are of interest.
One of the most frequent conservation end uses is translocation. That is returning plants to the wild, either as new populations or bolstering existing ones. In this video, we are going to learn how an ex situ collection can be used for a conservation translocation.
Translocations are about establishing viable populations of plants in the wild and ones that have the genetic diversity that allows them to adapt and persist when conditions at the translocations change.
The Western Australian Seed Centre have been conserving Grevillea calliantha with the long-term goal of restoring wild populations.
Well, Grevillea calliantha was listed as threatened way back in the mid-90s. So it's known from 6 small populations up from the Dandaragan area, which is about 150-200 kilometres north of Perth. Most of the populations were only known on road verges, and it was challenging to manage these populations in the long term. So translocations then became one of the most useful recovery actions we could do to make sure that this species had a chance of actually persisting in the long term in the wild.
Before commencement, the team needed to develop a translocation plan, which detailed the how and the why of the project. They also needed to review permits, approvals, hygiene risks, and exit strategies. Then they could start wild seed collections based on their decided sampling strategy.
So for a conservation seed collection, we want to make sure that we sample widely both within a population, but we also want to make sure that when we're sampling a species that we sample across the populations. And we want to make sure that that sampling is random. But for other purposes, maybe for plant breeding, you might want to make considered decisions when you're selecting for forms or flower types or those sorts of things. If you have populations that are quite disjunct, you then need to make a decision about whether you can mix those populations in your translocation. In the case of Grevillea calliantha, all of the populations occur in a relatively small area. It was decided that all of those populations could be mixed when a translocation is undertaken.
Once in storage, propagation methods can be tested and include vegetative or seed germination.
For rare species, often we don't have a lot of seed to be able to try lots of treatments in order to work out the optimum way of germinating something. So we might use surrogate species to work out our methods and then apply it to our threatened species so that we're not using up that limited seed source.
Trying to establish how best to germinate or propagate Grevillea calliantha was done through trial and error and also using background information that people already knew about how to germinate or how to propagate Grevilleas in general and other species that also occurred in the same family as well.
Once the living collection is established, reintroductions or translocations can occur, but that doesn't mean the extra collection is no longer required. Often, an experimental or staged approach is needed.
We've been trying different methods of establishment as we've gone. We don't go too large at any one step of the process, so that if for some reason things don't go right, we still have more seed and opportunities to come back and repeat that process in the future.
So after we had planted them, we installed a watering system to make sure that the plants could be watered over the first 1 or 2 summers. And then also we installed fences to ensure that they didn't get grazed by herbivores such as kangaroos and rabbits. The other important thing that we do when we put plants out there is we tag them. And we ensure that we record all the information that we already know about those individual plants, and then we can follow each individual plant at the translocation site. So we have actually had quite a reasonable number of plants survive, and they've produced flowers and seed. We've had very limited recruitment. We know that recruitment is probably not likely to happen in large numbers in the absence of fire. Some sites are doing better than others, so as we learn which sites are better for establishing Grevilleas, we're focusing more and more on those sites. And then we're thinking about, well, what are our next steps as well? So success comes in many forms. It's not just understanding whether the plants survive, but it's also other aspects of their ecology and their life cycle.
Conservation collections are a long-term commitment and need replenishing. Often, fresh collections will be sourced from the wild, but it isn't always an option.
In the case of Grevillea calliantha, some of our seed collections represent populations of the species that are now extinct in the wild. So in that instance, regeneration is really the only option. The next step in that translocation process will actually be to establish a seed orchard to bulk up seed of that species. But regeneration can pose problems for a long-lived perennial species that may take a long time to reach maturity or may require a very large area to grow an adequate number of plants. So in those instances, we would then try cuttings. So some plants can be successfully grown from cuttings. We can still get a good genetically representative sample to use for those translocations.
Translocation is one potential end use. The decisions at project commencement about intended use will inform everything that follow, including collection, propagation and storage methods. But there are always unexpected challenges and uses, which is why partnerships are so important.
Partnerships and collaborations are absolutely essential for starting and maintaining ex situ collections. Conservation often doesn't receive a lot of funding, and we require support from public and private corporations to support the work that we do. Organisations such as the Australian Seed Bank Partnership, which really put together a coordinated Australia-wide response to ex situ conservation, and ensuring then we also have the duplication of material across Australia. And then finally, for research using ex situ collections is often multidisciplinary and requires a great deal of expertise across a wide range of different areas. So including plant biologists, soil biologists, or even people who work with animals.
Detailed information about utilisation of ex situ germplasm is included in Chapter 15 of the Guidelines. The FloraBank Guidelines deal specifically with seed sourcing and ecological restoration. The Translocation Guidelines can be used to plan and undertake best practice translocation.